Hello, my name is Maeve Wilder. Thank you so much for joining me for this presentation on my Red Rocks campus floristic survey. The primary goal of my project was to collect and document all flowering plants on campus. I had been part of a project with the Denver Botanic Gardens last summer to do a floristic survey at their Chatfield location, so I had learned the necessary skills then. So for my survey area, I chose only to look at parts of campus that had been relatively undisturbed for the past 50 years, so like not including any of the landscaping around the rec center or any of the lawns around the buildings. This left me with an area of about 150,000 square meters that I surveyed over the course of the months between October of 2019 and April of 2020. So most of the habitat on campus has been developing since the main buildings were built in 1971. Habitat on campus is considered to be part of the shortgrass prairie biome, which extends from the mixed grass prairie in the Midwest all the way to the base of the Rockies. So this biome is dominated by grasses, as trees cannot really withstand the harsh disturbance regimes in the form of fires and grazing that this ecosystem has to endure. So 70 to 90% of the plants are going to be buffalo grass or grama grass. They have deep root systems that anchor the topsoil and make it so they can survive extreme temperatures and the harsh conditions they're used to. They also provide habitat for small burrowing animals such as prairie dogs, which we certainly see a lot of on campus. The seasonal creek that runs through campus gives us a small riparian area in which you'll see things like cottonwood trees and cattails, and also great horned owls nesting if you're lucky. I visited campus 3 times over the course of my project to survey and collect samples. I used a meander search method, which captures a greater diversity of species on campus and provides a more complete picture of what's actually present. So I was basically looking for any plant that was flowering. In the fall, I also collected seeding and fruiting plants. So when I found a plant, I would take a collection photo of it for identification purposes. The photos also had GPS location data attached to them, which would later be useful for the ID tags. And I would look around and make sure that there were other plants of that same species in the area, and if there were not, I would not collect the plant. So when I did collect a plant, I would make sure to get all parts that were essential for identification, such as flowers and leaves and parts of the root system. As soon as the samples were collected, they had to be processed for pressing to prevent any excessive wilting or browning. So this required arranging the plants on newspaper so that both the fronts and backs of leaves were showing and that essential parts of flowers were visible. For succulents and plants with woody stems, they had to be bisected so that they would lay flat and any goo on the inside of succulents had to be scraped out. So the plants were put between layers of newspaper and cardboard in a permanent press. At first, I did not have access to a professional permanent press, so I used OSB board and straps, and it worked just fine. The Colorado climate is dry enough that I didn't need a drying cabinet for the specimens to turn out pretty good. So I just left the press for at least a week with the plants inside of it, and it's best to check the specimen and change out the paper so that they dry fastest. Once the specimens had pressed, I was able to mount them each individually on their own piece of mounting paper. So this required finding the best parts of the specimen and putting a thin layer of glue on one side. I also kept a small portion of each sample for a tissue sample for any potential future DNA analysis that might need to happen with them. Once all the specimens were mounted, I was able to use them and their accompanying collection photos to ID the plants. So my original plan had been to go to the research department at the Denver Botanic Gardens and get some help IDing my plants from the volunteers in the herbarium. but that was not able to happen with the current pandemic, so I turned to Wildflowers of Colorado, who organized their site by flower color, which was very helpful, and I was able to ID the vast majority of the plants I'd collected. So I would find their scientific name and their family and all of their common names, and I would put that along with their location data and their habitat types on an ID tag, along with their collection number and the date they were collected. There were 4 species. This is one of them that I was not able to identify yet, but I am still working on that, and hopefully they will be forthcoming at some point soon. I also made note of some additional observed species that I saw during my survey, but that were not flowering, so I did not collect them. I know you can't really see them because of my video here, but there were quite a few non-natives and noxious weeds among those, as well as my collected species. So, the 2 most troubling invasives I saw on campus during my survey were Dipsacus phelonum, or common teasel, and Euphorbia myrsinites, or myrtle spurge. There have already been some efforts at removal for teasel on campus. This is sort of a rough estimate of the worst of the teasel populations on campus, so it is quite widespread, but it is being tackled. It is a B-list noxious weed in Colorado, but the myrtle spurge population is, I think, more of a pressing issue. Myrtle spurge is a List A noxious weed in Colorado, which means it has to be eradicated immediately and not allowed to reproduce. It is a very fast propagating and spreading plant that can tolerate poor soils and a dry climate, which makes it excellent at spreading in Colorado. It was originally from Southeast Europe and Asia Minor and was brought here as a decorative plant, and has since become a real problem and is outlawed, I guess you could say, in Colorado and Oregon. It has a sticky white sap that is an irritant to the eyes and skin, and it also causes severe gastric distress if you try to eat it. So the main population I found on a triangle of land between 2 forks of the gully where the creek is. I also found a few individuals on the west side of the creek. I did not find any on the east side of the creek, but that does not mean they are not there and should not be wiped out as soon as possible. Invasives seemed to gather around the creek where there's the most moisture and the greatest disturbance regimes, which are going to give them room to take root and push out native species. So while the teasel population is already being tackled, the myrtle spurge population also needs to be looked after because it's a problem when it starts to take over. So the final herbarium collection has 24 mounted plant samples, 20 of which are identified as of now, with their accompanying ID information and tissue samples. These will be housed on Red Rock's campus. I also produced a comprehensive list of all the species I found and paired them with their collection photos. These resources will be available to anyone in the future who wants to look more into the flora on Red Rock's campus, or who wants to continue this floristic survey through the late spring and summer. I had a really good time with this project. Thank you so much for watching.